pleasure to be with friends again. Thank you for all coming to Toronto. We hope you enjoy our city. So I was asked to speak on GI, GR, GL, update on methods, and these are the people who are supporting us, and so I've had some consulting fees from General Mills, and I've received a salary from GI Labs, and there's a few other, I have no grant research support, a few other honoraria. My wife and I are owners and employees of those two contract research organizations. And we mitigate our bias by always disclosing our conflicts and not owning any IP and divesting myself of all financial interests in any companies with which my company or myself deal with. So what I hope we'll be able to do today, and my learning objectives are for you to be able to state the difference between glycemic response, glycemic index, the glycemic load, and to state why the 210 ISO method for determining the GI foods is superior to previous methods. So we have this question about what is a glycemic response. People are interested. There's interest in the regulatory bodies. Companies want to make foods with low glycemic responses. So how do we measure that? And how do we compare things? How do we determine the effect? And I'm going to be using data from this interlab study, which a number of you here in the room participated in 2003. It was published, where I sent foods around to 7 labs, experienced labs around the world, and these are the responses that we got. So there's been several attempts or methods of talking about glycemic responses, and a common term, the most recent, is the term glycemic response, defined by the DRI as the effects of carbohydrate-containing foods on blood glucose concentration during the time course of digestion. And of course, the problem with this term is it's very vague, this definition. I don't think it's very suitable at all for regulatory purposes, at least defined in that way, because you can really say anything you want about it. And so if we just look at the responses to potato and barley, the concentration of glucose, barley could be anywhere from 24% less than potato to 13% greater than potato, depending on what you want to say. If you look at the increment, it could be up to nearly 90% reduced. The peak concentration, 22%. The peak rise is 56%. There's different, this is a vague term, and there are lots of different metrics and measurements you could use. You might want to ask, well, are the measures correlated at least with each other? And so these are the results of 7 centers comparing one food to another for different metrics. So here's the difference between barley and potato for the 7 labs. The peak rise correlated with 45-minute concentration, not particularly well correlated. Peak rise very well correlated with incremental AUC for barley versus potato. Total AUC, incremental AUC, not particularly well correlated. The problem here is that if you look at a different comparison of different foods, these correlations fall apart, and in fact, there's even a negative correlation between total area under the curve and incremental area. So the larger the difference in incremental area, the smaller the difference in total area. So I'm not sure glycemic response is a very useful term, at least defined the way it currently is. So what about glycemic load? And this was introduced by the Willett group in '97, and it was defined as the glycemic index times the grams of carbohydrate divided by 100, adjusted for energy intake. And these were the famous papers showing that a low glycemic load reduced risk for developing diabetes, both in men and women. And I think this paper did an awful lot to bring GI and GL up into the public interest. I have a concern about the use of glycemic load from a clinical point of view because I don't think the meaning is really very well understood. My conception of the meaning in all the popular books, and I've even been co-authors of them, is that it is the glycemic response elicited by the glycemic load grams of glucose. So this is, if you multiply the GI times the grams, the glycemic load of 1 apple is 8.8, glycemic load of 2 apples is 17.6. And so there's 2 ways you can reduce the glycemic load. Easiest way is to eat less carbohydrate. However, the actual original meaning, and the way the Harvard group talk about it, is that it's adjusted for energy. So it's the glycemic load per 1,000 calories. And in this case, the glycemic load of these 2 servings of apples is the same. So that I think we got to work on that. Another problem is how we determine the glycemic load. Of course, you can calculate it from the glycemic index, which assumes you can measure the glycemic index, and also the grams of carbohydrate, available carbohydrate. Many foods, though, you might want to have a glycemic load for, you can't measure the GI because there's not enough carbohydrate. So what do you do there? Well, you can directly measure it by looking at the glycemic response of a serving of food, area under the curve of 30, for example. If you measured your glucose, 50 grams is 100, so you read it off the graph and it's 15. And that's been done that way. But I think we need to realize the dose response curve for carbohydrate is not linear, it's curved, and you need to take that into consideration when you're planning your experiments. And in fact, this would overestimate the glycemic load. In fact, it's about 10. Glycemic index was the first kind of proposed scheme for classifying carbohydrates. We introduced in 1981. The reason we had equal carbohydrates was because we kind of more or less assumed people would supposed to be eating a moderately high carbohydrate diet. The question wasn't how much carbohydrate, the question was what kind. And diabetes were exchange lists, and it assumed that 15 grams of carb from one food was the same as 15 from another. So that's why we were comparing them based on equal amounts of carbohydrate. And in 2003, we did an interlab study. This was really suggested by Hesty Foster from South Africa to see whether the method was any good. And in fact, the conclusion of that study was that the between lab standard deviation was about 9. And frankly, I was a little bit disappointed with that. That seemed rather high. And a number of our critics have considered that to be rather high. And I thought, well, these foods all needed cooking. And perhaps if we, the cooking might have affected it, different cooking. And so we did another study where we fed ready-to-eat foods. And this involved 28 labs. Some of them were rather inexperienced. Some of those inexperienced labs did very well, and some of the very experienced labs did very poorly. I'm not saying any more, but we had the same conclusion, standard deviation 9. And so people felt, and the regulatory agencies felt this was rather large, and it is a bit large. And so they proposed that we shouldn't use GI and we should use glycemic response instead to classify for labeling for regulatory purposes. So I thought it might be of interest to use those data from that study to assess how precise glycemic response is. So over here, we have expressed, we've expressed the data for potato, rice, spaghetti, and barley as a percentage of the response for bread, area under the curve. And here is the glycemic index of these 4 foods. These are the means and 95% confidence intervals for the 5 labs involved. You can immediately see, for example, not all the 5 labs showed that barley was significantly less than bread. There's 100% for bread. Whereas there's the GI of bread at 70, that's high. All of the labs had a GI, 95% confidence interval less than bread. And in fact, we can see standard deviation was twice that for GI. Well, and you'll say, well, these are higher means, so if we take coefficient of variation, it's still significantly higher. So glycemic response is not more precise than GI. In fact, it's less precise than GI from exactly the same data. Now, all of those 2 interlab studies use the original GI method from 1981. And we now have a new method, 2010, and the essential features of the method are the same in terms of morning amount of carbohydrate, the blood sampling schedule, area calculation, and so on. But the new method has several new procedures and criteria which are required to be met. And the purpose of these, so this was using 10 subjects, the reference coefficient of variation, that's within individual variation of subjects' responses to the reference food, exclusion of outliers, 2 fasting samples, and a good analytical method with a low CV. These were all meant to improve the precision and accuracy of the method. But in fact, we didn't really know whether that actually would occur until quite recently. This was a study done by one of our clients, presented at FASEB this year. involving myself and our lab and Sydney and a lab in France, where we tested 6 foods. And these are the means and the 95% confidence intervals of the GI values for these foods. And the mean standard deviation is 4.7. Half of that what it was the original studies. Well, I didn't really think that was quite right, because we don't know how these labs, different labs perform differently. And maybe these are great labs. Well, they are great labs. But fortunately, the same 3 labs participated in the second interlab study, and these are the means and 95% confidence intervals of their results, and they averaged an average standard deviation of 5.9 between the labs. So they actually are performing better than average, but we still have a 20% lower standard deviation and a 30% lower CV. We have a CV now less than 10%, less than 8%. That's better than many medical tests done in the hospital. So, what is the difference? Maybe I went too fast. Can you tell me the difference between GR, GI, GL? Just to remind you, glycemic response compares equal servings of foods. The methods and the endpoints are not defined. Glycemic load is the amount of glucose required to elicit the same glycemic response as a serving of food. It accounts for the source and amount of carbohydrate, at least in the terms of the way it's popularly thought to mean. But in fact, the meaning depends on whether energy is adjusted or not. And most of the data, my understanding, all the epidemiological studies supporting this, they adjust for energy. Glycemic index is the glycemic index of a food relative to glucose, controlling for available carbohydrate. It's precisely defined methodology and precisely defined endpoint. So why is the new method superior? Because it includes the various procedures and processes to improve accuracy and precision. So I'd conclude that the glycemic index yields more precise results in glycemic response, and that the ISO method may be more precise. I think this certainly could use some more investigation. But the current estimate is that mean between lab standard deviation is less than 5. So thank you.
Energy adjustment is never at the food level. It's actually on the entire diet level. So in other words, the glycemic load can all be calculated based on simple products of glycemic index and available carbohydrate. So which is have a direct interpretation of what you mentioned about the glycemic response and whatnot, with the exception of a common reference that's glucose. Now, if you look at those original papers at Harvard where they do the energy adjustment, they're actually using the so-called residual methods that based on the food frequency questionnaire, they allow them to calculate the total energy intake of the diet rather than each specific food items based on the serving size. But one can also calculate, obviously, each glycemic load at a food level, adjusted for energy at the food level also. But it is important to make the distinction between the two.
Okay, I'm still not clear what the interpretation that is, but thank you.
Because you take into account the substitution effects. In other words, if you interpret the glycemic load in the context of a whole diet, so you are not only interpreting the effects of glycemia per se, but also there's some built-in substitution effects. In other words, the foods could have contribute energy from other macronutrients such as fat or proteins might be substituted in the context of the whole diet. And that's the idea, adjusting for total energy intake, which is somewhat different from at the food level, physiologically.
So if you could please identify yourself when you speak, so that because we're videotaping.
I'm Simon Liu.
Thank you.
Once upon a time at Harvard, and 20 years ago, did that study with glycemic load and coronary event in the nurse's house study.
Am I calling? Who's asking? Jenny had her hand up first.
Yes, Jenny, please. Jenny,
Just a simple comment. The way I explained glycemic load to my students, who are relatively innocent of all the controversy, I just say that the glycemic load is the carbohydrate content adjusted for its glycemic index. So think of it in grams, think of it in the same way you think of carbohydrate, but its GI will adjust that value. So if the GI was 50, it's actually half. So it seems to work.
Right.
Yep.
Your thing?
Yeah, Nate Maniszewski from Untitled. Just a quick question on the thanks, Tom. It was a great presentation, and I agree that there's been a lot of steps forward, and with the ISO method, it's great. Just a technical question on the glycemic response versus glycemic index comparison. What was the metric from glycemic response that you used in that comparison?
I expressed the results of the foods as a percentage of that for bread, and that would be very similar to what the Health Canada proposed guideline was, where you compared one food to a similar food you could use instead, and it had to have at least a 20% difference in the incremental end of the curve in order to get a claim.
Tom, a couple of questions. This is Frank Sachs from Harvard. Do you always test, say, 10 people? So those same, just so I'm clear about your current method, so you take 10 people and you feed them the reference, which is what, white bread now, or glucose?
Glucose or white bread can be used.
Or white bread.
You
and
have
then,
to adjust the white bread value to the glucose scale.
And then you feed them, what, in a sequence? The test food, the same amount of carbohydrate in the test food. Do you feed so these 10, is it always 10 people, or do you?
Okay, so the reference food is repeated at least twice, and if we have a largish number, if we're doing 3 or 4 things, we'll do it 3 times, and we usually do it at the beginning, the middle, the end of the series. It's the same 10 people. If we have, if a client has a specific product and we think the response is going to be less, we would do a power calculation and use more subjects if they wish to determine, to find a difference which is significant for something which is less than about 30%. If you're just, remember, the GI is not really meant to be necessarily comparing foods. It's meant to be an analytical measure which gives us a result for a number for a food. And of course, if you use more subjects, the precision of that number becomes better. 10 is a number which was kind of conceived of. Sometimes we use more. Sometimes we don't use less.
Yeah, because I remember seeing some of the raw data, and there's quite a lot of variation and outliers. So I'm not surprised that if you can apply an outlier criteria, you'd get a better result, and maybe that is the way to go. But I have another question on those 10 people. So, who are those 10 people anyway that you use? In other words, what sorts of folks are they? Are they old, young? What's their fasting glucose? That sort of thing.
Well, we have a very loose criteria. We don't have a restriction on body weight, and we have an age, just basically 18 to 75. We've demonstrated that age, sex, glycemic response status, whether you have diabetes or not, none of these things impact the value you get. So the glycemic response is, of course, higher in people who have, for example, if you're over 40, the glycemic response is higher than if you're under 40. But the glycemic index, we had a study where we did about 40 people in each of those 2 groups, and it was exactly the same, even though the glycemic response is about 20% higher, significantly higher in the over 40s. So again, GI is not a glycemic response. It's a relative to glucose in the same person. And the things which make the glycemic response high would be the same for glucose and for the food, with a few exceptions. But those are relatively rare, and I think they need to be looked at.
Great. You just answered my next question. Thanks.
Jeff has a question.
So for example, if you're lactose intolerant, you will not have a glycemic response to milk. We know that.
Jeff?
Well, it's very nice to be here, and thank you very much for that talk, Tom. One of the issues you mentioned for assessment of glycemic load was the difficulty of definition, and perhaps method of expression as well, which those involved in food analysis and so on often call the mode of expression. But you compare GI with GL, and yet there are all these different modes of expression. Can you be sure that GI is always more precise than GL, independently of all those modes of expression?
Can I be sure that GI is always what?
More, it always comes up with a greater precision.
Well, I didn't compare the precision of glycemic index and glycemic load because they're basically the same. I compared it to glycemic response. Yeah. I imagine if we looked at different comparisons, we may not get, there's variation. I mean, the glycemic responses are inherently very variable. So, and my point was when people complain about that, that is the nature of the beast. And whether you can express it as glycemic index, glycemic response, you're always going to be more variable. Glycemic index is going to be, on average, less variable because the reference is done 2 or 3 times, so we have a better indication of the subject's true response to that thing. So if you did your glycemic response, if I had done the bread twice and then compared it, I would have expected to get fairly similar results for glycemic response. But that's not part of the method. In fact, the methods aren't given for that, and that's not typically what's done when people are comparing 2 things.
Just one more point, if I may. That is, when you look at between laboratory variance, you look at the variance in the mean values.
That's right. Those standard deviations were the standard deviations of
rather than the
means.
source of variation.
Rather than the source of variation?
Yeah.
Yeah.
Because the figure that you calculate can be divided into 2 sources. One is variation within laboratory, which is contributing to the values that you get, and the other is the interlaboratory variation. Now, I've analysed the first collaboration that you did. In fact, one that you let me analyse many years ago, but only this year I went back to the paper and reanalysed them from the summary data. And you can get very low standard deviations for the between laboratory once you've taken out the analytical variation. So it is only around about 5%, even for that study.
Okay, nice to hear it. But I'm not sure if in practical terms, you're going to send your food to a lab, it's going to give you a result.
Furio has a question?
Just a comment, more than a question. It's related to the accuracy of the data that we put on food labeling or food labels. So we have not to forget that labeling is regulated by a very large, we'll say, net of rules, which also allow to put energy or nutrient value based on food tables without any reference to the actual food. So I would like to see your opinion, or maybe also to reason a little on what we can allow in food labeling in terms of dietary fiber content, for example, or nutrient content, with respect to the accuracy which is required for quality or carbohydrate index, such as the glycemic index. So it would be surprising to see that not always when we say that a food contains 3 grams of fiber, it really contains 3 grams of fiber. And also which kind of fiber we are defining with that number, since also there the methods are not so completely aligned.
Yeah, so the Canadian Food Inspection Agency rules regarding carbohydrate are that if you pull off a food and any one value can be 50% different than the labeled value, and the average of a number of them can be 20% difference. And even the original GI with a standard of 9 would be highly likely to meet those criteria if you were going to go and do some testing. So I do take your point. Foods are biological things and they vary.